Okay, welcome back, everybody. So we are now going to move to our next session. This is single-use systems in biopharmaceutical industry. Our presenter is Isso Bayala, instructor of biotechnology from Quincy College. Isso, the floor is yours. Thank you so much.
Hi, everybody. Can you see my screen?
Yes, it's good.
Okay, good. Thank you. So I'm going to be talking about single-use systems in the biopharmaceutical industry. You all have been using single-use system, whether it's plastic bags in the store or other bags. So those are single-use systems. But in the biotech industry, the biopharmaceutical industry, it is regulated. It's regulated under regulations called GMP. So GMP are regulation put in place by government to make sure certain companies make products that are safe for consumers. So it doesn't apply to all the companies. So it's mainly companies involved in food, drug, cosmetic, medical devices. Those companies have to follow GMP. So because of that, the equipment, the reagents, the supplies used by those companies must be in conformity with the GMP regulations. So in this talk, I'm gonna go over the single-use system, how it started. Then I'm going to talk about the integration, where in which area of biomanufacturing, biopharmaceutical industry it is used. Then I'm going to talk about the advantages and also the disadvantages. The main disadvantage is everybody should know that it's because they are mainly plastic, they are not biodegradable. So that's the main disadvantage of the single-use system. Then I'm going to talk about validations. Then I'm going to talk also about the future of single use in biomanufacturing. Then they're going to be, I'm going to give a summary of all of that. So in this slide, so I'm talking about how the single-use system have started. So they have been around as long as the biopharmaceutical industry also has been around. So you all are familiar with plastic pipettes, petri dishes, tea flasks, plastic LMAF flasks, roller bottles. Jim was talking about roller bottles a few minutes ago. And then we all use lab scale filters. Then certain labs use the membrane for diafiltration and concentration, TFF membranes. So all those are products that involve single-use systems. In recent years, there have been a surge in the use of single-use system because of improvement made in the quality of those products. So, for example, the extractables. The problem with extractable has been dealt with properly because of regulation. So extractables are compounds that come off the containers. So when you make your product, maybe buffer media, and you store them in a plastic bag or a container, if the material comes off the container, comes off and mixed with your product, that's materials called extractable. So at the beginning, there were a lot of problems with those compounds. And because of the regulation, GMP regulations, the companies have worked hard to solve that problem. So they use a material that is not extractable. And they are compatible with most products, for example, compatible with a certain pH, so that they don't come off, they don't melt and they don't mix with the product. Then you have also a problem of leachables, so that has been dealt with. The leachables are molecules or compounds that end up in your product that initially are not intended to be there. So, for example, there is an overlap between leachables and extractables. Sometimes, depending on the time, the holding time, so the time you store your product in a container, a molecule may form and they become a new product that mixes with your original product, but that new product actually is not intended to be there. It's not desirable. It can also, so these are called extractables, leachables. There is also what is called secondary leachables, which are, for example, those material that are used in the formulation. If you use buffers or you use additives to store your final product, those products may react and form a new product that can affect the quality of the original product. So those are leachables, and the companies have dealt with that because of GMP regulations. Another reason why there has been a surge in the single-use system is because of an economic advantage. So many companies are now involved in producing those single-use systems, and so the price have dropped, and so many people can afford them compared to originally. The other advantage is it's much easier to use them because, for example, I remember we used to wash petri dishes because they were out of glass, and we autoclave them and reuse them. Today, you can save a bunch of time by just buying petri dishes that are plastic, but they are sterile and you can use them right away, and you minimize the contamination. So those are the main advantages why there is a surge in the single-use systems. Any question? So if you have a question, just ask it, because I'm going to cover the upstream, downstream, single-use system involving all those parts, and I may be able to answer some of the questions. I may not be able to answer some of them. So, but if you have a question, please just ask. So this slide tells us about the history of single-use systems. So before the 1990s, industry companies involved in manufacturing drug products, they had facilities that were only allowed to make only one product. So you make one product, and so you can see on the first picture on the left here. So look at the facility. It looks like a real factory. So these are stainless steel bioreactors, and with a pipe, so they are part of the building. So actually, it's a part of the building. You can't separate the manufacturing facility from the building itself. So because during that time, the regulations were that if you're making those products, you have to make only one product. When you finish, you clean up, you set up. There is what is called a setup time, so a turnover. So you autoclave, you test, and you start a new batch, and make sure there is no leftover of the previous batch in the new product you're making. If you get that, then there's a violation against GMP. So in the 1990s, we start seeing those rules have relaxed a little bit, and companies were allowed to make different products as long as they don't do them at the same time. So you finish one type of product, you clean up, you set up the facility, and you do the changeover. Then you bring a new raw material and you start making your product. But in the 2000s, because of the single-use systems that have start coming to the market, so we have seen that industry has started using more and more of those because they are convenient to store buffers. They use bags to store buffers, to store media, or even to take samples, including hospitals where they store blood samples. Then you also have seen the way bioreactors. The way bioreactors have been around for a longer time than the bioreactors themselves. So it's a bioreactor. But the size is much smaller, but it's usually bigger than a tea flask or a shake flask or a conical. So then you can grow your cells there and give them the optimal conditions to grow. So today, there is what is called modular portable flex factories. So what it means is you have those single-use systems that are not a part of the building. You can actually bring them in piece by piece and set them up in a room. So you're gonna have bioreactor, you're gonna have a mixing system, you're gonna have filtration systems, and you're gonna have purification systems. They are all separated from each other, but you can actually install them in the same room. Then you start making your products. So in this slide, I'm showing most of the companies, the big companies involved in making single-use systems. So you can see Thermo Fisher is one of them. Sartorius is one of them too. Make a lot of bioreactor weigh bags, all those parts. There is a Charter, there is a ASI, Corning. Corning makes a lot of single-use vessels. You have NUNC, Cytiva is one of them. They are based here in Massachusetts, and they make a lot of single-use system from way bioreactor to bioreactors all the way to 2,000 liters. Then you have also Applicon. Applicon does both traditional and the single-use system. You have Belco, and you have also Sigma. Millipore Sigma is also, they have a lot of facilities here in Massachusetts. And they have one facility here that makes only single-use systems. So I'm gonna show you later a picture of that facility. So this is an example of a 3-liter bioreactor, single-use bioreactor made by Millipore. So you can actually grow your cells in this one just one time and throw it away. This is just maybe expensive for most labs, but industry can easily buy those and use them. On the right, you have another single-use bioreactor made by Eppendorf. So you can also grow your cells there. You can add media, you can add gases, you can take samples, you can mix it. There is actually an impaler that can actually, you can use a motor to run it, or some of them are magnetic. You put them on a magnetic plate, and you can spin the blade to mix, or to create a mixing system. Sartorius, for example, has a complete system. They have a base to mix the bioreactor for the impaler. And that base is connected to a power supply. And here is the vessel with many, many tubings. Those tubings, you can use them for, usually all the tubing come from the top are called addition port. So addition line. So you can use them to add things in there. You can also take samples and to measure pH, count cells, and add acid or base. You can do all of that. But this is a small bioreactor, 2-liter bioreactor, a single use. Okay, next slide is a roller barrel. Roller barrels are still used. People don't like to use them, but some cells grow very well in the roller barrels. The roller barrels are similar to flasks. They are usually used to grow adherent cells, not in suspension, because they are more surface for the cells to bind to compared to the flask, because the cells bind to the inner surface of the flask of the barrel, and you put a little bit of media only at the bottom. And there is a belt that rotates the barrel. So because you rotate the barrels, all the cells, if you put them at the optimum speed, so all the cells are gonna have access to the media over the time, so they're not gonna starve. So you adjust the speed and you use a little bit of media, and that's enough to grow them. And you can see the caps, vented caps, so you can use that for aeration, give them the gas they need. The other thing is, the problem is to harvest. To harvest is not easy. You have to trypsinize them, and there is a risk of contamination. But some companies don't have a choice, because the cell line they're using grow very well in the roller barrel. They don't grow in suspension or in other containers. But other companies also have a robot to harvest, so which is more efficient, but it's costly for most companies. So in this table, I'm showing the markets. So the projected market of the single-use system. You can see this projection was made in 2018. So you can see that it is projected that the market will reach 40 billion in 2023 for the single-use system, 20 billion for upstream processing, and 20 billion for the downstream processing. And for the stainless steel, it's around 30 billion, so it's much lower. So you can see that the market for the single-use systems is increasing rapidly because of convenience, and we're gonna talk about that later. So this is the same projection here, but so it's detailed. So based on the different part, the kind of single-use system used here. For example, you have tubings. The tubings use a lot. For companies that use them, they use about companies involved in biomanufacturing, and they use about 90% of tubings. Tubings are used a lot. I'm gonna show you later how they are used for connection, for transfer, all those parts. So, and for TFF, you use all those tubings. So they are used a lot. Then you have also bags that are used a lot, and you have connectors that also are used a lot. You have filters that are used a lot too, and we're going to talk about why filters are used a lot, because they are convenient, and some of the products, for example, you're making media, some of the media cannot be autoclaved because of the protein will denature, and if you use a filter, you can remove contaminant without damaging the product of interest in your media. Or in your media, or in your harvest. So Jim and Margie were talking about growing cells. So you start with a Cryova. The Cryova is just usually a small container, 1 to 2 ml, where the cells are stored at a cold temperature. So because that container is made of material that can handle very low temperature, from -80 to -150 degrees C, and it doesn't crack, and it's used a lot for cryopreservation, including embryos. So even those containers are single-use systems. So you use them one time. Once you take them out, you empty them, and they become trash. So they are single-use systems. Then the next thing, you are tubings. Anytime you have your products, whether it's media, cells, buffer, you want to transfer it. And if you have single-use system, you want to use tubings. On the market now, there are different type of tubing. The tubings you want, if you're using a welder, are called weldable tubings. Most of them are called C-flex tubings. So those are plastic that can be welded and keep your product sterile. I'm going to show you the welder later. Then you have other supplies that are used a lot in most labs, like the Eppendorf tubes or the microcentrifuge tubes. They are all single-use systems. Then you have the conicals. Some of them are vented, some are not vented. So mostly the vented one are used for cell culture. Before, we didn't have the vented one, so we have only the non-vented one, and we use them to grow cells, so we have to keep the cap loose. That also can create a risk of contamination. And now with the vented one, you can keep the cap closed, but then you can also, the cells are gonna have access to the gases needed because of the filter inside. And you're gonna also prevent contamination. So it's an improvement in that regard. So these are barrels used for storage. So you can use them for media, use them for buffers. Usually they have a plastic bag inside those barrels. So, and you can see the tubing at the bottom that you can pull at the bottom, and you can drain or you can draw some of the solution inside there using the tubing. So the bag inside are single-use bag. The barrels are not single-use. You take a new bag, you put the bag inside, and you put your product in there, and your product is safe, and you can actually drain your product. Put on a scale. Usually, with a large volume, you use a scale, not the volume, not a cylinder, because if you want to draw 100 liters, you just use the scale, and you put a container on a scale, and you draw 100 kilograms. So that's 100 liters. So these are also used. These containers are from Millipore, and many companies have them, and they use them in buffer and media prep area. So you put your buffer, and they have wheels, and you prepare your buffers, you put them in those bags, and you can push them to the area where they are needed for protein purification, or even for, if it's a media, you push them to the cell culture area where they are used for the bioreactors. These are petri dishes, single-use systems, been around for a long time. Everybody has been using those. The petri dishes are convenient. They come sterile, they are gamma irradiated. And they are mostly plastic now because the glass ones are fading away because of the convenience. So use them for cell culture, all of that. So next slide. So these are again a flask. You have filters, syringe filters, and you have also Erlenmeyer flask. So two types of Erlenmeyer flask. So the one on top here from Cell Treats, Chemglass also has those. You buy them, they are vented, they are single use. You can grow your cells in there, and usually it's just one time use. And because the cups are vented and the risk of contamination is relatively low. And these one at the bottom are much better because you don't really have to open the cup. You can, after you can open the cup inside the biosafety cabinet, you can inoculate your cells, input your media, inoculate, and you close it, you put it in an incubator, and you grow the cells. You can, once it's time to inoculate a whey bioreactor or another flask, you can just use the long tubing. The short tubing is just a vent tube, so because of the filter. So the longer tubing is used for transfer. So you can weld that to another container and you can pump your culture into the next vessel. So what you're gonna use is called a stereo welder. So the stereo welder, I'm gonna show you the stereo welder in a minute. So again, like Maggie was talking about the Eppendorf bioreactors. Now they have what they are called samplers. So the samplers, you can actually, they are autoclavable. You can attach the sampler when you build the bioreactor. You attach the sampler to one of the sampling ports. So then you autoclave the whole thing. And when it's time to sample, you just put a syringe in there. And you can draw your sample and go for testing. So it make it much safer, prevent contamination. And when you draw your samples, you can use another syringe and push it and push your culture that is in the line back to the bioreactor using the other line with the filter. You just push, create a pressure, and so the cells don't stay in the line. They go back into the bioreactor to have access to the media so they don't die. So, and the good thing is this support is autoclavable. And for schools, labs, we use them so many times. You can always autoclave them and reuse them. Okay, so these are also single-use systems. These are T-flask and IPF flask. So the IPF flask are like T-flask, except they are built together. They are in a stack, one on top of another. So what it does is it just gives you more space for the cells to grow. Usually these are for cells that are adherent, so they not suspension cells. So they need a surface to bind themselves, so like the T-flask. So this is like a bunch of T-flasks on top of each other to create more space in one single container. And to harvest, you can use, you will be able to use a scraper would be difficult to use. You can use a scraper with the T-flask. And these ones, to usually use the trypsin. You can also use trypsin with the T-flask. So two ways of harvesting the cells. The next is a single-use bioburden filters. So those are used a lot in biomanufacturing for environmental monitoring. So if you have buffers, you have media, you have your water for purification, you want to test those samples to make sure they don't contain unwanted microorganisms. So you pass them through a filter like this. And this is filter, the filter inside becomes, the whole filter is gamma irradiated. So once you pass your sample through the filter and you remove the filter and you put the filter on the surface of a petri dish and you incubate. So this is a membrane filter. This filter is so thin that when you sit it on the surface of media on a petri dish, the cells can actually reach the media and they can grow on the filter. So for example, this is a bunch of microorganisms that grow on this filter. So the sample, whichever was filtered, contain microorganisms. You only can see them when they grow and become many colonies. Then you can identify them. If you are in the food industry, you want to make sure none of them is salmonella, for example, because those are unwanted microorganisms you should test for, required by GMP, so they don't end up in your final product. So, so they are single use, and they come sterile and they are convenient. You can use them. So this manifold, the blue one here, is made by Millipore. And if you buy them, you can actually filter 3 samples at the same time. Next slide is a mixer. Before I talk about mixers, this is just an example of ingredient used to make a buffer, or it can be used to make a media. So you have, in industry, you're gonna have the ingredient and you're gonna have a recipe. This recipe can be on an SOP. And they ask you to make a media. So you don't necessarily have to make it, if you're using the single-use systems, you don't have to make it in a clean room. So you can use the recipe and weigh out your ingredient, put them in a container, in a barrel like that. The barrel is just a container, but there is a plastic bag you put inside. That plastic bag is called a liner. So the liner is this sterile plastic bag, is thick, it's so strong, and so it's open, the top is open. So you can put your ingredient in there and you put your water in there. Then you use this motor. This motor has a blade. You can lower it inside and you turn the motor on, you mix your product. So when you mix your products, so that's how you prepare your media or your buffer. So, but because it's in open area, it's not a sterile. Now you have to sterilize. To sterilize it, you're gonna use what is called a bag with a filter. So this bag, for example, has a filter here. So you take a tubing, the same tubing you were talking about, you take a tubing, you attach the tubing to the inlet of the filter. And you take the other end of the tubing, you put in the barrel, and you use the peristaltic pump to pump your product, your buffer or your media, through the filter inside the bag. So then you collect how much you want, and you clamp it. Then you detach the tubing, and this bag now gonna have the product that is sterile, the microorganism. So you don't have to do that in a clean room. It can be that in a regular lab or in a non-GMP facility. So then you get a sterile product. If it's a media that is sensitive to heat, so if you use this type of system, you won't affect the quality of your product, because the filter will just remove the microorganism and will not affect the quality of your product. So then you use the peristaltic pump. The peristaltic pump doesn't have to be sterile because the peristaltic doesn't come in contact with your product. So you just put the tubing around it. So this is an example of a bag that has a media in there, and the media has been filtered. So this media is considered sterile because it went through the filter.
So were you serious that we could ask a question during your talk?
Yes.
So I have a question for you. So I see this is all single use.
Yes.
And so I figured companies mix and match single use depending on the company and the product they're making. Or am I correct? And it's based on cost,
Oh
effectiveness.
yeah, that's correct. Yeah, but I'm not sure. I'm that's so. I'm not sure that that's a question. Yeah, the cost is something they take into consideration.
And Jim just said based on compliance. Okay. So I'm trying to see which one is most important. Compliance, cost, they're equal. It's
yeah, so
compliance.
the good thing is if you're gonna use a single-use system, the burden is on the company that make the single-use system to comply with the regulations. So they have to make a bag, for example, that is compatible with the GMP regulations. So they have to test it and validate it. So if you buy those products, you have to do minimum validation. So most of the validation are made by the producer of the systems. Is that your question?
So I think you did just answer my question. I could see if you were a company and you were developing a new product.
Yes.
And since you're developing a new product,
yes,
then maybe you would add in single use because it'd be easier or more cost effective.
Correct. That's where the many companies, especially the small companies, are using a single-use system because they don't have to they don't have to build a facility that is expensive. And they can move around. So if they can move from one facility to another area that is where the rent is lower, for example. So many companies, for example, we have a city here called Cambridge. That's where the rich companies can afford to rent, like the Pfizer's. But the small companies cannot rent that area because they don't have the capital yet. So they can, if you have a single-use system, you can just put in a truck and move it to the outskirts of the city, small city, and rent a place, and you can set up. It's much easier, flexible, and that's why. And because many companies also are involved in making the single-use systems, the prices are going down. There is a lot of competition now. The other problem that was there before was the compatibility of the part. So if you buy part from Cytiva, part from Cytiva, and maybe Cytiva is not around anymore, can you get another company that make products that are compatible with the system you have in place? Now, many companies making parts that are compatible with each other. So that problem also is going away. So it makes it just much easier. Any other question?
I think you answered my questions. And we educate our students on weigh bags and things like that. But I was just trying to get a feel for how single use is being introduced into the industry and all the parameters that were affecting its introduction into the industry. So, I'm just trying to get a feel for this.
Oh, okay. So I think I'm gonna give you more information as we go.
Okay.
So there's more information, and please feel free to ask if I don't answer your question. I will try if I know the answer. So these are mixers. So these are just, you see, they are mobile devices. They have, some of them are motors. Some of them have a motor that is separate on another device with an impeller. So you can actually, you make your product in the bag. Those those mixers have inside single-use bags. So only the bags are single-use. So you remove the bag, install a new bag, and you put a new product in there. And to mix, you can just use an impeller like that, a motor, and you lower the impeller in, and you turn the motor on, and you mix your product. To sterilize it, you pass it through a bag, through a filter, and that filter is attached to the bag. They come together, and they are all gamma irradiated. So the filter is sterile, except so you just have to pass it through the filter. Everything stays after the filter. Everything stays inside, and everything that goes inside the bag gonna be sterile. So the sterilization also has become easier, especially for large volumes, and also for products that are heat sensitive. We cannot autoclave, for example. So this is another mixer for media or buffer. So the only thing in there is if the holder is the same, you do a CIP, cleaning in place, but the bag is new. You just install a new bag that comes gamma irradiated. The bag, they come, they are the label. There is expiration date on the label, and there is a sign that they are sterile. They have passed the gamma irradiation test, so they are sterile. So the burden is on the company that make those bags to sterilize them before they sell them. Okay, so this is you can use a serial welder, or you can use a quick connect. So I have a video here. I'm gonna try to play it to show how you can connect without using a welder. So hopefully it's gonna work. So this is from Millipore uses a lot of this. So
Isso, you're not sharing the video if you think you are.
oh, I'm not sharing the video. Okay. Okay, can you see now?
Yes.
Don't forget to share computer sound.
Oh, the sound. The sound doesn't work. So I'm just gonna explain. Those are two tubings that have. So you wanna connect those two tubings, but there is a membrane on each side of the tubing. So when you pull, what you do, you align the two tubings so the membranes gonna sit on each other. So when the membranes sit on each other, then you pull the membrane, you pull the blue piece, so you just tear off the membrane. So then the sterile side of the two tubing gonna connect to each other. So then you push them against each other. So you align the membrane. So then the dirty part gonna be on top of each other. So then when you pull the blue, you pull the two membrane at the same time. Then now you're gonna leave the sterile side of each tubing connected to each other. Then you push it and one more click and you connect them. And you make a sterile connection that way without contamination.
fantastic. Thank you so much, Isso. This was great.
So is my time over?
It is, but if you want to take just a few moments to wrap up, I know it kind of stalled in a place where you were. I know you have a few more things to say, but you can definitely go ahead and take a couple of minutes. Yeah.
Okay, so let me just go to the. Can you still see my screen?
No, you'll have to reshare. You have stopped sharing.
The advantages of the advantages of the single-use system are the following. So you have less labor requirements, so it's easy to install, set up. So you have you eliminate the sterilization and the cleaning. So you reduce CIP, SIP, almost no SIP. So then the setup is the turnaround time is shorter. And so the holding time, so you reduce validation, and you also, so it's become flexible. So it's a flexible setup. Also, the space used is reduced, so you use less space. So that's the conclusion.
Thank you very much. This was very informative and very useful. I'm sure I will keep track of all the conversations and pass that on to you so that if there is more you can add, you can please go ahead and put that in chat as we go forward.
Okay, thank you.
Thank you.
you.